Hi guys, so I'm back with another video as promised on molecular and furthermore cytogenetics. So today we'll be starting with fish for fluorescence in situ hybridization. I'll be breaking up fish into multiple parts so that we take the topic slowly. I would first discuss in general, then I would go to the principle and the procedure, then I'll go to the test and show you some images of each type of molecular as well as cytogenetic technique. So I'll be starting with fish, and fish can be used in a lot of techniques. Mostly it's used in molecular diagnosis of blood cancers and some solid tumors like breast cancers. But similar techniques, keeping similar principles in mind, you will also study in not under the heading of molecular, but in say heading of immunofluorescence. Now, immunofluorescence staining has a very similar principle, if not the same. Of course, there's no RT-PCR in immunofluorescence. There's no increasing in copy number of the DNA, but there is antigen-antibody reaction and fluorescence involved. Here in fish, there is no particular antigen-antibody reaction involved. However, there is the role of fluorescence in fish also. So without wasting much time, what is fish? Today is that is what I'll be dealing with, and then slowly I'll also show you the procedure and the principle and the results of a fish. Okay. So another slide taken from the net. Fish is the basic element where you try to you take a chromosome or a DNA strand, you denature the DNA, and then you have a complementary amino acid sequencing strand of DNA provided in the kit by the person who the companies like Thermo Fisher, etc., which make kits and probes for fish. And these complementary strands having complementary amino acids to the DNA that is there in the host or the patient, they have fluorescent labeled antibodies on them, and this strand when will go and attach to this strand, and there'll be a multiplication of both the strands, will lead to fluorescent light being emitted, which will be captured by software and be displayed on your microscope, or not captured by the software where there are no software available. There will be a reaction when this complementary kit complementary DNA labeled by with a fluorescence probe present in the kit goes and attaches itself to the strand, which is mutated in the human body, and like my pink nail polish, a green or an orange signal will be emitted, and that is what you see in a microscope which has filters to observe these fluorescent images. So this is the a very layman way of what fish is, how we observe fish in the microscope, right? So again, there are to continue before I show you this image, that is why things where you have to attach a synthetically manufactured antibody or a synthetically manufactured DNA probe to a known DNA, to a diseased human patient DNA probe, it is quite logical and understandable that this will only happen when we know कि कौन सी अमाइनो एसिड डिफेक्ट हम स्टडी करना चाह रहे हैं. फॉर इंस्टेंस इफ आई नो कि मुझे ट्रांसलोकेशन 922 ही पढ़नी है, तो इस क्रोमोसोम में अगर ट्रांसलोकेशन 922 पढ़नी है, तो मुझे जो कॉम्प्लीमेंट्री डीएनए बनाना होगा मार्केट में, जिसमें मैं फ्लोरेसेंस लगाऊंगी, एक डाई लगाऊंगी, वो भी 922 की ही होनी चाहिए. Unknown reactions padna is not is impossible in fish. Fish, you need to know what mutation you want to study. Otherwise, you cannot use fish as a technique for studying that mutation. You cannot find out new mutations. You need to know that मुझे HER2 new ही पढ़ना है breast cancer में. Then only will I be able to use fish as a technique. So fish can only be used against a known mutation that you want to study. So we have a DNA probe. Through splicing techniques, we create gaps in the disease DNA. We add probes with labeled fluorescence dye. and then we go ahead and we try to denature again the two combined. This is another way of doing fish, but this is how another way the machine can process the fish. The end result will be the same, and then we again denature the probes, we amplify it through to a thermal cycler, they again go and reattach and give you fluorescence. So these lighted points, this orange, yellow, red signal, green signal, this is how fish appears on a blue background which is dark, and these bluish color, this is DAPI. This is how the unstained cells which do not have that mutation will appear under a microscope. So this is the normal DNA and RNA structure. You need to know this. CGAU, this is the RNA structure. CGAT in the DNA, there is uracil and RNA and thiamine and DNA. We need to know this because if the mutation is say at the point A in particular chromosome, then the probe that will develop will also have the mutation at the same point complementary to A and will have a fluorescent dye attached. So A attaches to T. So the probe should have a T over here. If mutation produces an A, then the probe should have a T so that they attach, they bind, and a fluorescent signal is produced. Okay. So it's a study of chromosomes in detection of genetic abnormalities. It combines conventional cytogenetics with molecular genetics in hematology, pathology, and constitutional genetics. And the things that you need is fluorescent probes, and these fluorescent probes bind to complementary parts of the nucleic acid in a DNA. So special regions there are which give fluorescent dye, and they determine abnormalities of specific segment, and you should know what abnormality you want to study. So there are two types of fishes. One is fluorescent in situ hybridization where you use fluorescent dye, and then there is simple in situ hybridization, each very commonly done with P16 where we are not using a fluorescent dye. Okay, but the technique is same of in situ hybridization. In situ means within the chromosome. Hybridization means we are trying to pair up all complementary probes to the mutated site in the patient. Fish can be of many types. There can be quantitative fish, mostly to study the length of the telomere in cancers and aging cells. Fusion signal fish, Philadelphia chromosome, CML. Multiplex fish, 20, this is known as chromosome painting. The entire chromosome looks like a rainbow. And this is done, as a replacement to the black and white karyotyping. But still, it has not replaced karyotyping completely. Multilocus fish where more than one mutation need to be studied. Alpha centromeric fish, this depends on the centromeric probe where it is placed and the exact location of the mutation. So the common signals that you get is gray, green, blue, and yellow. Blue is the background signal, that of the DAPI mostly. Yellow is the fusion signal, and orange, green, or red, green are the break apart signals, which I'll be dealing with in the consequent lectures, what they are actually. So again, we have a target DNA and we have a market produced probe. When we want to do fish in any specimen, we take a fresh specimen, bone marrow, blood, whatever tissue, we culture it, we increase the number of cells, then we lyse the cells. Then what we get is the nucleic acid material, the nuclear material. We put the patient's DNA through denaturation so that both the DNA strands which are intertwined together, they separate. Now, in the same patients, two DNA strands which were twined, they separate. Now, when they separate, we have the synthetic market manufactured complementary DNA with labeled these two dots that you see, they are labeled fluorescence. Now, if there is the mutation that we want to study, suppose in translocation 922, if it is present, then this probe is meant for translocation 922. It has the mutation synthetically in it. This will go and attach to the two separated probes to one of them and give a fluorescence, and that fluorescence we study. Just before doing this step, we the steps of hybridization, before this what we do is we put the sample in a thermal cycler so that there is not just one copy or 50 copy of a normal patient's DNA, there are 5,000 copies of the same patient's DNA, so that the intensity of the fluorescence that you get will be higher. Now, this can be done when we are doing RT-PCR. Supposing you are not doing, this is part of molecular. Supposing you just want to observe in fish, you just want to simply see whatever number of cells you have cultured them, you have spread it on the slide, then you don't need to put it in a thermal cycler, the sample. You just, read the number of signals, say in 100 cells, you are getting 50 signals, 50 cells showing this kind of mutation, where this market available 922 mutated DNA probe went and attached to the autologous, also the complementary site on this DNA fragment, and this fluorescence gave a light, and that light you could see in the microscope. So this goes both the ways. It can go into the molecular spectrum, or directly a simple pathologist my fluorescent microscopy can also be done. So before hybridization, the DNA probe is labeled. This is available in the market. The labeled DNA probe is denatured to a single stranded DNA. This single stranded DNA, to this single, if there's mutation present, the market available probe will attach and give a fluorescence. And you see these are all red is the fluorescent dye, the color, and this green is the market available probe. And you see these two DNA strands are separating. The probe is attaching to the complementary faces, and on the microscope you see a red signal. Okay, so there are two pairs of chromosomes. If mutation is there, you'll see two red signals on attachment. So probe can is complementary to the target sequence, fluorescently labeled 20 to 100 base pair, and specific mutation, specific to a particular mutation. It's a specific region. It's not an unknown mutation. And what can you study through this? You can study duplications, deletions, translocations, etc. Types of probes, they may attach to the center of the centromere, that is known as metacentric. One of them is metacentric. The types of centromeric probes can be submetacentric, acrocentric, and telocentric. Then when I see the show you the procedure, I'll tell you one complementary fluorescent label probe attaches to the mutation, the other one attaches to a centromere or a telomere, just to help you in counting the number of chromosomes that have denatured. That, and I'll tell you how it is done subsequently, so don't get confused. Then they can be whole chromosomes probes, and what we commonly use are locus specific probes. Means this probe is for a particular mutation. So gene specific probe only where the allele is mutated. Centromeric probe will go and attach to the centromere. Telomeric probe will attach to the ends, and the whole chromosome might be painted with a probe. Means multiple, the entire complementary probe has yellow light emitting out of it. Okay. What do we target with fish? RNA within cells, DNA in metaphases, and DNA in interfaces.